And now I'd like to introduce our next keynote speaker, Dr. Goodell, who will speak on the functional roles of DNMT3A. Margaret Peggy Goodell is professor and chair of the Department of Molecular and Cellular Biology and director of the Stem Cells and Regenerative Medicine Center at Baylor College of Medicine in Houston, Texas. Goodell's research is focused on the mechanisms that regulate hematopoietic stem cells and their dysregulation in malignancies, particularly DNA methyltransferase 3A, or DNMT3A. Goodell was elected to the National Academy of Medicine in 2019. She is a recipient of the Donald Metcalf Award from the International Society of Experimental Hematology in 2023, the Tobias Award of the International Society for Stem Cell Research in 2020, and the Dameshek Prize from the American Society of Hematology in 2012, and the Edith and Peter O'Donnell Award in Medicine from TAMIST in 2011. Goodell is chair of the Scientific Advisory Board of the Keystone Symposia and is a member of their board of directors. She currently serves on the editorial boards of Cell, Stem Cell, and Cancer Cell, and has served on the editorial board of PLOS Biology, and as an associate editor of Blood. Goodell directs a laboratory of about 15 trainees. So please join me in welcoming Dr. Peggy Goodell to the stage.
All right. It's a pleasure to be here. I'm going to I have a lot to talk about. I'm going to try not to go too crazy on the time, so I've got a little timer here. Okay. I thought I would try to give kind of a big arc of how we're thinking about DNMT3A and what we're doing in the lab. And for us, it really all started with the blood system and the question of how decisions in a stem cell are regulated. And this is really what I found in my lab on it. We were trying to understand what genes control the decisions that a stem cell makes to either make more stem cells or go on and differentiate. And we had done a number of studies looking at how genes were differentially expressed during that decision process. And we literally just stumbled upon DNMT3A. It was one of many genes that were upregulated in stem cells and not in differentiated cells. As you can see here, these are two stem cell populations on the far left and the differentiated cells on the right. And there were a bunch of other genes that were in this category of expression, but we thought this was particularly interesting and for a number of reasons, it being an epigenetic regulator, and then somebody at Baylor happened to have the knockout mouse, and so it was just a convenient model to study at the time. So the way that we initially studied it is we got these knockout mice, and let's see if this works. Yes, it's gonna work. There you go. You can see the pointer, right? Yes, excellent, okay. So we took these knockout mice and we crossed them to our favorite Cree, and then we purified the stem cells and we transplanted them to recipients, and then we look in the peripheral blood of these recipients over time for what proportion of blood cells are produced from the knockout cells. And we can actually then take the stem cells from these mice and retransplant them in and look at their activity in multiple experiments over time. And really, I'm just going to try to very, very briefly summarize quite a lot of data from a bunch of different papers. But the bottom line for these very initial experiments is that we found that when we transplanted these knockout cells into new mice, we found a lot more stem cells were produced. And it doesn't really matter what method we're using here. There's different ways to purify stem cells. But if you look at this population here, it's very rare. These are the hematopoietic stem cells in the blood, in the bone marrow. And after the knockout, you see they're almost tenfold increased in number. Okay, so that was a pretty dramatic phenotype. So you get more knockout stem cells, so they seem to self-renew, make more stem cells better. But how much of an advantage of this? And so we did this crazy experiment where we took bone marrow from those transplants recipients and purified stem cells from those, and then retransplanted them into another mouse, and then into another mouse, and then into another mouse. And we did this basically every 4 to 6 months to look at how much those stem cells could expand over all of these different transplants. And if you just do this with normal wild-type stem cells, they will engraft in the new mice only about 3 times, but we kept doing this with the knockout cells. In fact, we did it 11 rounds of transplantation. And what we found is, in what you're looking at here, is their ability to differentiate. And you've got transplant numbers across the bottom here. And every time we're transplanting 200 purified cells, every single time. And what you can see is that after about 3 rounds of transplantation, the differentiation capacity of those pure stem cells completely disappears. But if you look at your ability to get more stem cells out of those mice, you maintain that number after every transplant. And this is, each dot is representing the number of stem cells from a different mouse. But what you can see is that the average pretty much maintains the same level over all of these periods, over all these transplants. So we like to think of it as these stem cells being immortalized. And we never really found a limit on their ability to make more stem cells, but they couldn't really differentiate anymore. And an important feature is that these stem cells look almost exactly the same as the stem cells from a wild normal mouse. They don't seem to be cancerous or anything at this point. They just look like normal cells. They've acquired this ability to go on and on forever, but they can't differentiate and make other blood cells. So this led us to this concept that DNMT3A is really a gatekeeper for differentiation, that it's really important for permitting. Normally, in the normal wild-type situation, it's important for permitting differentiation to occur. In the blood system, no other gene that's been described has quite the same role for differentiation, for permitting differentiation in the blood. And we view this as a progressive phenotype that's enhanced through cell division. So you see it sort of mildly, like initially the stem cells can differentiate reasonably well, and then the more you transplant them, the more those cells divide, the less they can differentiate. So the phenotype progresses, and you see more and more methylation, DNA methylation loss as you transplant them. So these are correlated phenotypes that are enhanced over when you push the cells to divide. So how does DNMT3A loss lead to increased self-renewal and ultimately to cancer when it's lost in adults? That's still largely unknown. I'm happy to talk about ideas about that. I'll touch on that a little bit, but it's not a big focus. What I do want to mention, we'll come back to DNA methylation in the last half, is that we, through the process of studying DNA methylation differences in the DNMT3A knockout situation, we discovered this genome feature we call canyons. Canyons are kind of like CPG islands, but they're really big CPG islands, so they're big regions that have very low levels of DNA methylation. And what we found is that DNMT3A, one of its main jobs is specifically at these regions to maintain the methylation. And so what you can see here, this is a DNA methylation track in wild-type hematopoietic stem cells. And each of these sticks is the amount of methylation at that. And we've sequenced over it like 30 times on average, so we have a good sense of the proportion of methylation at these CPGs across it. So high methylation, low methylation right over this gene. This is a canyon because it's quite big. And when you look at the DNMT3A knockout cells, they don't have any DNMT3A, we've really eliminated it. You can see this canyon has gotten bigger. So the canyons get bigger, so that means that's where DNMT3A is normally active. So they get bigger, and we also see less methylation at some other regions that are usually like enhancer regions that are controlling expression of this gene from a distance. So these are the two areas that we think DNMT3A has a particularly important role. So does that affect gene expression? So what I'm going to show you is actually from double knockout, knocking out DNMT3A and 3B, which is a more severe phenotype, but it illustrates the point and how we think about this. What we wanted to know is, we're knocking it out in the stem cell, but how does that affect gene expression in the downstream, the differentiated cells? And so we compared gene expression in wild-type stem cells and wild-type B cells, okay, one differentiated cell type. And then we mapped the expression of genes that were differential. And what you can see here in blue, these are the wild-type stem cells, and in green is wild-type B cells. So these genes are downregulated normally in the B cells, okay? So what happens in the double knockouts? Well, if you look at the double knockout stem cells, you see these stem cell genes, and normally stem cell genes, are expressed at even a higher level, so they look like even better stem cells from the gene expression perspective. And in the B cells derived from those genes, they're supposed to be B cells, but you still see those genes expressed. So they have kind of a stem cell genetic phenotype, if you will, even though they are supposed to be differentiated cells. So this is one of the concepts that underlies the way that we think about things, is that even though the cells are differentiated, and they kind of look like B cells, they're really only halfway there. So they really inadequately repress the stem cell program when they do go on and differentiate. Okay, so the cellular mechanisms that lead to this enhanced self-renewal. Stem cells show less DNA methylation around important genes that control that. The differentiated cells don't completely suppress that self-renewal program. We think there's many small effects that lead to this ability to keep making more and more stem cells. And this really is a paradigm that we use to think about a lot of other aspects of DNMT3A function. I'm not sure it necessarily applies in all tissues. It's kind of our bias, and we're always trying to check our biases when we're looking and thinking about other tissues and other systems. This is kind of what we use to think about things. So the other thing I want to emphasize in the blood is that DNMT3A, its main role is really in the stem cells and in the very early parts of differentiation. It doesn't seem to play a major role in the downstream cells. However, it's kind of like a hit and run. If you don't have it in the stem cells, you see phenotypes in those downstream cells, but that's because it's missing upstream. And so if it's not there to do its methylation job in the stem cells, then it creates an aberrant phenotype from all of these downstream cells. So again, this is sort of part of the concept. You're looking at the downstream phenotype, but the business end of what's really happening is in the stem cell, at least in the blood. Okay, so DNMT3A promotes differentiation in blood stem cells, and also we realized that there was old literature that even in embryonic stem cells it has this same role. So if that's the case, how much is this paradigm true in other stem cell systems? We don't know that broadly, but we have looked at that a little bit in the generation of fat cells, of adipocytes, as Kate mentioned earlier today, and this was driven by a postdoc in the lab who's now at Novartis. And this was published a few years ago. I'm really just going to summarize what she showed, but really to try to give you the analogy to the blood system that we just talked about. So we realized when we made these heterozygous knockout mice, we were originally just studying the blood, and we had left some of them on the shelf quite a long time. We realized that some of them got quite obese. And you can see in here quite easily, these were about probably 6 months old, and these mice were much bigger. It was really kind of the accident of leaving them in the cage too long. Like the boss doesn't want that to happen because it's an expensive thing to do. Anyway, they appeared to be obese, and so we looked at this much more systematically. And again, just to give you a few snippets of this, these were showing you the difference in the weight gain of wild-type male mice and heterozygous knockout using the heterozygous null allele, fed on a normal diet over time. And you can see until about 4 months, they pretty much look the same in the cage, and then they really diverge, and the mutant mice continue to get quite large, and in fact, it really affects their mobility and everything after time. So why are they obese? Kate sort of referred to this a little bit as well. So if you ask neuroscientists, or at least the ones that I've asked, they say it's all hormonal control, all of weight control is all in the brain, right? And what we wondered if that was really necessarily the case, because the adipocytes are also generated over time through adipocyte progenitors in the fat. So Ayala decided to look at this really from both angles, and she found that when she put the mice in a cage, this controlled system where you are monitoring how much they eat and how much they're active, are they not moving as much, so they're not burning as many calories, are they eating too much, what's going on? We found that actually they do have greater food intake. I don't know if you could call them hyperphagic. I don't, where do you call something hyperphagic rather than just eating too much, like I do most of the time. But they definitely have an increased food intake during the day and even in the evenings, and whether it's light or dark cycle. But they also have a leptin resistance. So even when they've had more food and there's really no difference in their physical activity that we could measure, there's no shutdown of that system that tells them to stop eating and to stop accumulating fat. So what about the fat tissue? So there is some central control, we think. But what about the fat tissue? So the dynamics of the way that they really increase in obesity over time, it sort of reminded us of what we see in the blood, in that you get this massive increase in stem cells, especially after transplantation. So we wondered whether there was a stem and progenitor cell origin in the adipocytes similar to the blood system. And so Ayala went through and looked at a number of different things, both in the adipocytes, in the fat tissue of the mice, both brown and white adipocytes, but also the progenitor system, which I'll show you a little bit. The short of it is that there's an increased size of adipocytes, so those are the fully differentiated cells, so they are accumulating more fat, more lipids in themselves, and they're getting larger. But to what extent are those changes cell intrinsic, so really controlled by that program? And so she took cell lines for this, both a white adipocyte progenitor cell line and a brown progenitor cell line, and knocked out DNMT3A in those, and then would compare the ability of those to differentiate into adipocytes and have normal sort of adipocyte-like function with the knockouts. And she used, both white and brown adipocyte cell lines. So first of all, when she just knocked out 3A and just looked at their proliferation, both of these models showed increased proliferation, as you can see by the red line, over time. So they're already behaving differently just by knocking out DNMT3A. She also saw differences in their lipid uptake, so there were actually fewer lipid droplets after you stimulate them. You can see by the red line again. And if you look at their ability to break down lipids, which is also a stimulated system, she saw slower release of fatty acids. So when we looked at this also at the gene expression program, it really looks like, although again they sort of seem to differentiate into adipocytes, they really fail to fully upregulate the gene expression program. So again, they're sort of halfway between being adipocytes and adipocyte progenitors. And what you can sort of see here, hopefully, is if you look at differentiation genes here in these two different models, you can see the red here is these differentiated knockout cells, and you can see the program is really down-regulated to where you expect it to be compared to the wild-type cells. And you can look at a bunch of different genes, and you can see that the same thing is true in both of these different model systems. So the adipocyte progenitor cells, again, like the blood, they're sort of getting partway to the differentiation, but it's a really incomplete maturation process. So what about the in vivo phenotype? These were just cell lines. There's ways to get adipocyte progenitors. This was all new to us, but they're basically mesenchymal progenitors, and you go through a flow cytometry step-by-step process, and you can get these, and you can then quantify the number of progenitors, the number of differentiated cells, and we did single-cell analysis on this. And again, I'm just going to summarize quite a lot of data here, but when we did single-cell analysis, we could really classify these stem and progenitor cells, these mesenchymal stem cells that reside in fat tissue, and then we saw these primed preadipocytes and then committed preadipocytes. You actually lose the fully differentiated adipocytes because they're very fragile, they're big, they're full of lipids, and they just kind of disappear in this whole thing. But what we could see, that we did lots of different single-cell analysis, and we could see that in wild-type progenitors, you go through this canonical differentiation process, and you can also go through an inflammatory differentiation process under certain contexts. And the wild-type and the heterozygous knockout tissue behaved very differently. So we looked at this at 2 time points, and if you look at this just first in young mice, you can see the heterozygous mice, the wild-type mice, the cells are kind of differentiating. These are the progenitors here at the bottom left, and then they go through this differentiation process up to the right. This is single-cell analysis. And the heterozygous knockout cells in the young animals have a very different behavior, with more of these cells sort of accumulating in a more earlier progenitor state. And there's also more of these that we would characterize as having an inflammatory process going on by a number of different inflammatory markers. And if you look at old mice, this is kind of, again, even the wild-type mice are sort of backed up in this early progenitor state, and they have this higher level of inflammation. And again, you can see that the heterozygous knockout cells, mouse tissue, is also similarly backed up and highly inflamed. So it's like the heterozygous mice, even at a young age, are taking on a phenotype more of wild-type older mice, even though they're younger. Just as more gene expression and inflammatory program that we see. So if we think of this in terms of like a TBRS phenotype in the mouse, we definitely have these behavioral differences. And we have this leptin insensitivity, and we have this abnormal adipocyte and inflammatory phenotype. And we think really all of these together are contributing to this obesity phenotype that we see in the mice. And importantly, like we see in the blood, we think these adipocytes, the stem and progenitor cells show aberrant differentiation. Okay, so there's aberrant stem cell differentiation in embryonic stem cells, hematopoietic stem cells, adipocytes, and a 2010 paper suggested that neural stem cells in the brain also show this. In fact, I had read this paper early on. There were a number of things that kind of didn't really make sense about this paper, but the more I studied this in other systems, I realized this original paper, this very, very early paper, was probably right, and they saw made some of these same observations. Okay, so to what extent can this sort of blockage of differentiation explain other TBRS phenotypes? I would say we don't really know, but again, thinking about it from the hematopoietic system, if you have a situation where you're loss or less DNMT3A builds up this stem cell population, you have a larger pool of stem and progenitor cells. Ultimately, that also means you have more differentiated cells. As long as there's not a complete block, you actually increase the pool, you increase the number of differentiated cells that come out of that pool. And so this is a way of making a larger tissue, if you like, okay? It may explain some of the growth phenotypes. This is just speculation, but I think it's worth putting this sort of stem cell paradigm, into our minds and thinking about the way other tissues in TBRS patients are growing. Why are they so much taller and so forth, and the bone phenotypes that were talked about earlier as well today. And again, I want to think about that the methylation alterations that are set in those stem and progenitor cells are manifest to the downstream cells. So you can look at all you want in the downstream cells, but at least with this paradigm that I'm talking about, the business end is happening in those stem and progenitor cells. And the reason that I'm emphasizing this is that this view, if it's correct in many tissues, is really relevant to any therapeutic approaches. You can put DNMT3A back or do whatever you want in the differentiated cells, but if you really want to make a long-term alteration, you're going to have to think about how to alter the progenitor cells and hope that that is transmitted then down to the differentiated populations. Okay. So in the last few minutes, I want to switch to mechanisms and how is DNMT3A functioning in these different tissues. So I told you that it's critical for differentiation, so what is it really doing? We know that it's involved in de novo DNA methylation at CpG dinucleotides, and we and other labs have suggested that it's got some non-DNA methylation activities, histone recruitment, RNA splicing, and we also have data that it's involved in establishing the infrastructure for higher order chromatin through its DNA methylation activity. So I wanted to think about the protein and the gene, the functional domains and what those domains are doing. And again, I'm just going to touch on all of this. So what we knew as of just a couple of years ago, I'm going to talk about the N-terminus at the end of this, but is we have a methyltransferase domain, we have this ADD domain, and we have this PWWP domain. And different functions have been mapped to these. And H3K4 is this reader domain for this ADD is H3K4 reader domain, and the PWWP reads the H3K36 status. So I wanted to sort of touch on all these domains except for the ADD domain. But you remember from the mutation maps that Kate showed us earlier, we see mutations really clustered in all of these domains and a little bit outside, but they pile up in these domains. Okay, so this again is a DNA methylation map. You'll remember your whole genome is basically completely covered by CpG, by DNA methylation, except for a few regions like CpG islands and these canyons that I referred to. And these regions where you don't have methylation are usually marked by different histone marks. So here is HVK27 methylation sitting in the middle of this canyon, and in other places where you have low methylation, you have H3K4 trimethylation, the activating mark instead of the repressing mark. So as we've discussed, DNMT3A, DNA methylation is established in development by 3A and 3B, maintained by DNMT1, and high DNA methylation can repress genes, at least that's our paradigm. So what happens when you knock out DNMT3A and you look at the DNA methylation? As I showed you in that blow-up picture, you lose DNA methylation, but specifically at regions. And now this is really zoomed out. And again, this is a DNMT3A knockout over a different gene. Here you can see two canyons, and again, they're larger when you lose DNMT3A. One of the things I want you to note is that overall, the DNA methylation does not look much different. Really doesn't look much different across this whole region of the genome, except at these few sites. And again, DNMT3A's job is really very focused in the way that we think about it. So you see loss of DNA methylation at these two canyons and between them here. We also looked at methylation after we transplanted those cells 11 times, like I showed you, when they really increase their self-renewal. And the methylation, remarkably, still doesn't look that much different. So they've changed their properties. They still look like good stem cells. You continue to erode the methylation at these foci, but you don't really massively change the landscape. This was kind of a surprise to us. So again, you can see here, there's now even less methylation here between and at these canyons, and it looks a little bit more raggedy than it did after only 3 rounds of transplant, and now this is 11 rounds of transplant, but again, it doesn't look vastly different. This is just a zoomed out look at that, and what you can see is that most of the methylation changes are at the edges of where there's already low levels of methylation. So that's where DNMT3A is normally sitting and doing its job. Okay, the same thing basically happens in the adipocytes, and I'm going to run out of time, so I'm not going to go into this in too much detail, but what I just really want to show you is that in the, you can see that there's, in wild-type adipocytes and adipocyte progenitors, you can see high levels of methylation that change over the course of the year. And when you look at the heterozygous knockout, you have less methylation originally, and it really doesn't change. So again, the methylation pattern is just, it's not completely gone, but it's just not really doing what it should be, either at the early time point or at the later time point. Okay. What about non-methylation mechanisms? We published just earlier this year that DNMT3A seems to interact with the spliceosome and with spliceosome components, so we think that it's involved in regulating splicing, and we can see at certain genes here, this is an exon and 2 introns, and this is just one example. In knockout human stem cells, we now see that the splicing is really inefficient. So DNMT3A seems to be contributing to having efficient splicing, at least at certain introns. Okay. So what is DNMT3A doing? How is it playing its role here? And this is also something that I want to emphasize, because there's a little bit of confusion about DNMT3A's interaction with H3K36. And there's a lot of papers that suggest that it's intergenic, that it's binding with K36 dimethylation, but we think that's true, but it's also definitely interacting with trimethylation over the exons. And so if you look, this is just different regions, but there's DNMT3A that's found right across these exons. I thought I had a methylation loss image as well. Oh, right. So here, if you look at where you lose DNMT3A in different regions of the cells in mouse ES cells, here's DNA methylation in wild-type cells, and when you knock out DNMT3A across the exons, you lose DNA methylation. and this is DNMT3B, and you really don't lose that much methylation. So, okay, so I've talked a little bit about DNMT3A having a role in, obviously, its catalytic role in placing the methylation marks, the H3K4 trimethylation reader, and we think that it's also very much that through the PWWP domain, it's involved in coordinating gene expression through interactions with H3K36, reading those marks, both at the di and the trimethylation mark, and probably this is what's being involved in when we see aberrant splicing as well, because H3K36, it coats the exons, and it's important for proper splicing and proper gene expression, and so it's this domain of DNMT3A that's important for that. Okay, so I just want to finally touch on the N-terminus, and we published this also very recently, so I'm not going to tell you too much detail, but we wanted to know, really nobody had studied the N-terminus and had a function for this, so we knocked out specifically that unknown domain, and we found when we knock out just that long isoform, you could see actually the mice essentially behave like complete loss of function, and they actually die 3 weeks after birth, whereas if you eliminate just the shorter isoform, there's really no phenotype. And we showed that you can actually partially, flying through all these last things, you can actually partially rescue that phenotype by just putting that long isoform back in the brain using Nestin-Cree. And so here, the mice with lacking the long isoform completely, they die 3 weeks or so after birth, and when you put it back under Nestin-Cree, you can see you partially rescue at least the lethality. So this long isoform is particularly important in the brain. Okay, it's going to bivalent regions. I really don't want to take too much more time here, so I will fly through this. And what I want to emphasize is that this is like the third domain, the fourth domain of DNMT3A, the fourth regulatory domain of DNMT3A. And so the 3A, the long isoform, is essential for life. It's deployed to developmental genes, specifically where there's polycomb marks. We think it particularly interacts with polycomb 1, and that may bring in polycomb 2. But this is likely playing a big role in normal development and pathological states. And so this is now our fourth domain here. So we think this is really important for polycomb interaction, and those people who are really interested in epigenetics will realize you've got all three things going on here, the three key aspects of epigenetic regulation. So you have your catalytic domain here, and 3A is really being regulated to, or prevented from going to active sites by this H3K4 reader. It's coordinating with gene expression by being able to read K36 status, and it's also reading the polycomb state through the N-terminus. It may also interact with RNA. There's implications in the literature, and it's probably this N-terminus as well. Okay, yes, I wanted to remind everyone that the other overgrowth disorders are also linked to readers or writers of those same domains. Okay. Okay, some final thoughts. We think at least some of the phenotypes are stem cell associated, but other functions of DNMT3A are also relevant. The mutation impact may relate to where in the protein is and which specific function, whether it's the methyltransferase domain or interacting through the PWWP domain and so forth. That's one of the things we think about a lot. I really liked Kate's emphasis on the phenotype database and the importance of the high quality of that. I think that we need an equally high quality analysis and catalog of the gene function of the gene variants. You know, some of us who are working on multiple variants and really what they're doing at the functional level in terms of their potency and what other proteins they're interacting with and so forth. And I think those two married together will do even better than just having, you know, one great database. I'll stop there. Lots of great people have contributed to this work over time. Ayala, I mentioned, Jaime Reyes. He's also at Novartis. Tianpeng Gu and Grant Challen really started this all off with his original observations in the hematopoietic stem cells, and many other collaborators who aren't even mentioned on this slide. So thank you very much.